For the case of tertiary structure, as I told earlier, there are three different methods are available. One thing is that you can predict the tertiary structure by using homology modeling. Homology modeling, there are three different model types are available. One is homology modeling, the other one is fold recognition, the other one is ab initio model. Okay, so why there are three methods are present for this particular tertiary structure? This is because homology modeling will predict the structure of the protein by considering its structural similarity, that is homology. So if you want to predict any tertiary structure of any protein by using this homology modeling, then you might have any protein already available structure should have similarity greater than 30%. So if any protein already deposited in the database which will have 30%, more than 30% of similarity in sequence to that of our sequence of interest, then only you will have a good results of homology modeling. So homology, if you want to predict any structure through homology modeling, you should make sure that our protein of sequence will have 30% of similarity to the already available protein structure in the database. Database means it might be in the protein structure database, PDB. Okay, so in that case only this type of modeling will looks good. Then the second method, that is fold recognition. So if any protein will contain say around 10 to 30% of similarity, then you will go for this fold recognition or threading. It is called as threading or fold recognition. That means we will find the structure of protein on the basis of regional part. Say a part of the protein structure is taken from different proteins and they are stitched together. That is why it is called as threading. So that is what the method is known as fold recognition. We are going to identify the tertiary structure by recognizing or identifying different folds from different proteins. That is what fold recognition. And the third method, that is ab initio. So it is nothing but prediction of a protein from scratch. That means if you are not at all finding any proteins in the protein structure database having similarity with our protein of interest, then we will predict the whole protein structures by amino acid by amino acid. Each and every amino acids are considered, and by applying different parameters, we will predict the whole protein from the scratch. That is known as the ab initio model of protein prediction. Okay, so that is why we will have these three different modeling procedures. So now we'll see one by one. So first we will go for tertiary structure prediction by homology modeling. So there are so many servers are available for this homology modeling prediction. The most familiar tool which is available for this homology modeling is the server which is available from the Swiss model. Again, it is from XPASI. So now we will see this particular Swiss model server. So we will open the Swiss model server. So start modeling, and this is the page it will open. So now we can paste our sequence in this particular box as a fast format. So we have only the sequence. So to make the sequence into FASTA format, I will type the first line with the greater than symbol, that is the FASTA sequence. Then in the second line, I am pasting the protein of interest. So this is what the sequence which is recognized by the Swiss model server, and they will give the sequence of the protein. So again, you can see this, all the 399 amino acids are recognized for structure prediction. Okay, then if we know, I already told you, if you want to predict a structure in Swiss model, then we can give already available structure of the protein to match with this particular sequence and to give the structure. So we can do that by using this target template alignment, okay, or user template alignment. So if we know if we are having the structure of another protein, we can use this user template, or we can know the other protein structure, we can go for this target template alignment through the online. Or if he doesn't know anything, we want to simply provide the option to the Swiss model server. It can identify its own template and predict the structure of the protein. Then we will go to this build model option. So before that, you can give the name of the protein, and if you give, you can give your mail ID to which this particular result will be delivered. Okay, so we will give this topic and a mail ID. Then you can click this build model. So it will take time for prediction of the protein structure. Okay, so if we already submitted a structure, then you will get a mail, or once the work is completed, you will get a mail indicating the available structure in the Swiss model server. So previously I have actually submitted one Swiss model. So now we will see that. So you can see there is a Swiss model result. Okay, we can go for this particular. So once the protein structure is completed, you will get a results like this. So here you can see this, the protein structure is predicted, and this is what the sequence we have submitted, and the template which is available in the database on which this particular protein sequence is predicted is given here. You can see this part. This part is actually predicted on the basis of this particular protein, and this part is predicted based on this particular protein. So like this, we can identify how this particular protein structures are prepared. And if you want to download the predicted structure, you can use this particular download file option to download the structure. Okay, so this is how we can get the tertiary structure prediction from the Swiss model. Okay, so there are about 5 models usually predicted for each submissions. Okay, so this is just now today I have predicted it half an hour before this class, and if the work is finished, here we have submitted, so once the work is finished, you will get a mail indicating the completion of the server. So just now you can see this. So the server completed the prediction. Okay, so now you can see this, the homology model structures will not be loaded now. So it will be loaded one by one, so that we can identify so on what basis the tertiary structure of the K space 9 is predicted. But you can see this, even though the protein structure is predicted, only the initial part you can see now. So this part is actually predicted nicely, but here also the end, both end, N terminal as well as C terminals are predicted clearly, but in between this N terminal and C terminal, the structure is not predicted that much. Okay, so this shows that the protein doesn't have a clear template. Homology template is available in the databases. So this is what the information we might get, and we can use this N terminal or C terminal domains for docking studies once predicted through this particular homology prediction tools. Clear? So now, so similarly, just now we completed this Caspase 9, so that server link is also provided here. So you can see this Swiss model result. So even the templates are loaded here. 3 models are predicted for our protein of interest. So this is how it looks. It is explained already. So you can see this. This is what we saw now, Caspase 9.